Virtual Atlas of Neuron is a project to incorporate the expanding knowledge in the field of neuroscience to an open-source, interactive web app of Neuron. The end product of this project will be a 3D interactive neuron model with scientifically sound and peer-reviewed anatomical, molecular and functional data. This video summarizes the first phase of Atlas development. A typical neuron consists of a soma, dendrites, and axon. Traveling across the cell membrane of soma, one finds an amazing world of subcellular structures and organelles. Nucleus has a nuclear membrane containing multiple nuclear pores. These nuclear pores consist of outer cytoplasmic ring and inner nuclear ring, held together through spokes. The outer ring contains cytoplasmic particles and filaments. The inner ring is connected to the terminal ring through nuclear basket. A number of molecules including RNA pass through a central region. As we enter into the nucleus through nuclear pore, we come across chromatin that can be in different forms depending upon the stage of a cell cycle. Shown here are 30 nanometer DNA fibers whorled around histone proteins, forming nucleosomes. Nucleus also contains a nucleolus, smooth endoplasmic reticulum, Golgi apparatus, work of cytoskeleton, mitochondrion, and enter into the axon, we come across a fascinating system of axonal transport of molecules. Tubulin and F-actins make the roads, while kinesins and dyneins are the vehicles. At the end of axon, there can be one or more axonal terminals that make synaptic connections with a postsynaptic membrane. These terminals contain their own powerhouses: the mitochondria, vesicles, and subsynaptic ribososmus. The cell membrane of axon contains voltage-gated sodium channels that are concentrated at the nodes of Ranvier, which is the unmyelinated part. Whereas myelinated sheet is formed by oligodendrocytes in the central nervous system and Schwann cells in the peripheral nervous system. Within this structural framework, we will utilize the open source, large data sets to add molecules and functional details. For example, this open source, the 3D structure of tubulin, will be linked to tubulin molecules, shown in this virtual neuron. Another example shown here is the 3D structure of nucleosome core protein. This virtual atlas of Neuron will keep on adding the molecular data as it becomes available, and will serve as the most comprehensive virtual atlas.